Hello and welcome everyone to this podcast for genetics. In this podcast for genetics, in the past couple podcasts we have talked about chromosomal mutations. And remember, with chromosomal mutations, we started off with a euploid set of chromosomes. For humans, that would be a diploid set, 2 copies of every chromosome. But sometimes we can have situations occur where we end up with 1 extra copy of that chromosome to make a trisomy. So again, 46 chromosomes in humans, and a trisomy would have 47 chromosomes. Sometimes, remember, we talked about how we could lose a chromosome, in which case we would have a monosomy. So a total of 46 chromosomes. And in some cases, we might end up copying the whole genome one time, in which case you might get a triploid. And for humans, that would be 69 chromosomes total. And we talked about various other combinations as well. And at that point, it may have seemed logical that by increasing a whole chromosome or losing a whole chromosome or many chromosomes added, that since we're changing the expression essentially of thousands of genes, it's easy to imagine how that could cause a phenotype. But what I'm going to talk about today is this concept of gene mutations. And with gene mutations, we're going to take this same chromosomes here, but instead of adjusting the whole chromosome, we're going to adjust just one gene. In fact, not even just one gene, typically just one nucleotide. Changing one nucleotide in one way or another. And hopefully by the end of this, you'll see how changing just this one nucleotide can cause an altered phenotype. Sometimes this altered phenotype is beneficial. Sometimes this mutation can lead to an unhealthy situation, some kind of disease. Sometimes there's no effect at all. And sometimes, at the other extreme, it can be lethal. And we're going to talk about all of this as we move forward. Before we talk specifically about mutations, I want to talk about location of mutations. I'm going to break this up into 2 broad categories: a somatic mutation and a germline mutation. Remember that somatic cells include all of our cells in our body except the germline cells, except for those cells that make our gametes. So a somatic mutation is a mutation that occurs in a somatic cell. Likewise, a germline mutation is a mutation that occurs in a germline cell, like the spermatogonium or the oogonium, one of these cells that helps make gametes. We might think of somatic mutations as occurring due to a mitotic error, and a germline mutation we might think of as a meiotic error. But it also could be due to a mitotic error. Because a somatic mutation will occur in one of the many trillions of cells, somatic cells in a body, the effect will be localized to that tissue. So for instance, in this lovely drawing of a human, if there was a mutation here in the heart, say, only the heart would be affected. None of the other cells would have that mutation. So somatic cells affect individual that gained the mutation. But remember, it is tissue specific. For instance, if this individual received a mutation in a gene that was important for heart function, and that mutation occurred in the heart cell, that would affect this individual's heart. That mutation wouldn't be found anywhere else in the body. However, if in the heart, a gene important in pancreatic function was mutated, it wouldn't have any effect at all. Only genes that are important in that specific tissue, if mutated, will have an effect. We may be walking around with all kinds of mutations in various parts of our body, but if it's not occurring in the tissue where it functions in, that gene functions in, then it doesn't matter. And because of that, somatic mutations are not heritable. Now, on the other hand, meiotic errors, or germline mutations, do not typically affect the individual where the mutation occurred. If I received a mutation in my germline that affected a heart gene, that's not going to bother me at all, because it's in my germline. However, I will be able to pass that mutation on to my kids, and now my kids will have that mutation, and it will be in their heart tissue, so it can cause a problem for my kids. Because of that, we say that germline mutations are heritable, meaning progeny are affected. Also should be noted that a mutation in a person's germline cells, that mutation will be in every single cell of the next generation or of their progeny. So somatic mutations are typically not good within the individual, but don't affect the progeny, whereas germline mutations are not typically bad for the individual, but do affect that next generation. So there's lots of different ways we could classify a gene mutation. One way we can classify a gene mutation is by the mutation's effect on the DNA or the gene that we're interested in. We can do this because a mutation alters or changes the DNA sequence. This can change the protein that was originally encoded by this gene. Throughout this podcast, I'm going to use these two terms quite frequently, and they are wild type and mutant. A mutant is any cell or any organism that has an altered sequence in a particular gene. So let's put DNA sequence change in a gene. Wild type, these are organisms that have no mutations. And when we say wild type, we don't necessarily mean that that organism doesn't have a single mutation anywhere. We mean it doesn't have a mutation in the gene we're studying. Just like a mutant doesn't have a mutation in every single gene, only in the gene we're interested in for that particular study. So we will use wild type and mutant quite a bit in this podcast and in future podcasts. Classifying gene mutations based on the effect on the DNA, we can call them base substitutions. We can call them insertions or deletions. And lastly, the third one we will talk about are these expanding nucleotide repeats. We'll come back to in a moment how these different kinds of changes in the DNA can have an effect on the organism. But for now, let's just talk about what it's doing to the DNA. Let's start with base substitutions. So just as it sounds, a base substitution substitutes one nucleotide for a different nucleotide. Base substitutions are either transitions or transversions. A transition is either changing a purine to a purine or a pyrimidine to a different pyrimidine. So for instance, a purine to a purine would be an adenine to a guanine, or vice versa, a guanine to an adenine. Remember, our purines are G's and A's here. A pyrimidine to a pyrimidine would be changes such as a cytosine to a thymine, or a thymine to a cytosine. Transversions are a little different. They can either be a purine to a pyrimidine, or they could be a pyrimidine to a purine. So purine to a pyrimidine, that would be like a adenine to a cytosine. It could also be a guanine to a cytosine, or a guanine to a thymine. Likewise, the pyrimidines to purines could be to a guanine. They could also be a thymine to a adenine, or a thymine to a guanine. It turns out that transversions are less common and transitions are a little bit more common. The consequences of these changes we'll talk about in a little bit. Next, let's talk about insertions and deletions. Don't get these confused with duplications and deletions at the chromosomal level. These will be insertions or deletions of single nucleotide bases, or sometimes it could be more, a couple or 3 or 4 nucleotides. All right, so insertions, let's go and write down that definition here. And that is simply adding or subtracting one or more nucleotides. We will focus primarily on the addition of one or the subtraction of one. To consider the effect of this, because you might think just adding one nucleotide, big deal, but it can have a huge effect. So let's talk a little bit about this now. If the original DNA, before it was mutated, the wild type DNA, was CGT, TAG, CCC, TGG. And of course, the sequence would be much larger than this. But in this very brief sample here, we can see that this DNA would code for 4 codons here. Previously, when we talked about substitutions, we might have been just talking about changing this T to a C, in which case you would still have 4 codons here, but this one mutation here will change this one codon to perhaps a different amino acid. And we'll talk about that again on the next whiteboard or two. But what I really want to talk about here is what happens if we have an insertion or a deletion. So let's look at a deletion here. Let's go CGT, and this time let's say instead of substituting the T, we're going to delete it. So it's no longer going to be there. So I'll put a dash there just so we can remember that. And I'll make it a different color here as well. So now we have AG, C, CC, T, G, G. Now, drawn like this, it doesn't look like too much of a problem, but what happens is we still have this codon, our first codon. Our next codon, however, is not TAG anymore. It is AGC. And this is going to code a completely different code amino acid than TAG. And then CCT will be the next one. And again, that next one is much different than CCC. And then GG, and then whatever is the next amino acids that come along here. We call this a frame shift. It's called that because every codon after the deletion will produce a different codon. These can have devastating effects on the gene that these occur in. Insertions are very similar. So again, this was a deletion here. Let's look at insertions. Insertions, CGT, and this time, instead of a deletion or substitution, let's add a G. And now the T is still there because it wasn't deleted. T, A, G, C, 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 T, G, G. Again, this codon is the same, but now we're going to create a new codon that's going to make a different protein or different amino acid. And the GCC is going to be a different codon. CTG will be G, and the next two amino acids that I don't have written there would be the next one. So again, this is also a frame shift because every position past the insertion will create a new codon and thus code for a different amino acid, or it might code for a stop codon. Again, these can have devastating effects. Next, let's talk about the expanding repeats. Your book will list several examples of these, but I'm just going to talk about one. I'm going to talk about the one linked to Huntington's disease. But in general, expanding repeats are a set of nucleotides that are repeated. Many examples will repeat a set of 3 nucleotides, so an additional codon. In the case of Huntington's disease, we'll have a series of repeats of C, A, and G at the very end of that gene. This codes for glutamine. So what happens is that you will have a series of these at the end of the Huntington gene, so you have a series of glutamines. Turns out most of us have these repeats, and between 1 and 37, typically no problem. But with Huntington's disease, if you go 38 or more, you will be more likely to have a problem. And there is a proportional response to the amount of nucleotides. If you had, say, 100 of these at the end, that's going to be more problematic than if you had 38 or 39. So the next thing I'd like to talk about are the way we can classify gene mutations based on their functional effects. And I'm going to focus on substitutions here. Not every substitution is equal. And so there are 4 different ways I'm going to talk about the effects of a substitution. But for all of these, I'm going to talk about this one sequence here, where the non-template portion of the DNA is GAA, and the template portion is CTT. When this is transcribed, it will make an RNA that is GAA. And it will make an amino acid that is glutamic acid. Glutamic acid has a negative charge on it. The first mutation I want to talk about is when that substitution, the first mutation I want to talk about is a silent mutation. A silent mutation is when you substitute one nucleotide and it still produces the same amino acid. So in this case, we're changing this part of the codon. We're going from an AT to a GC. This will make an RNA that is GAG. Let's write RNA over here so we don't forget. And let's write amino acid here so we don't forget that. So this GAC here, even though you change the sequence, will still make glutamic acid. So we call it silent because it has no effect on the amino acid that is made. Next, I would like to talk about a missense mutation. The missense mutation is still just changing one of the nucleotides, but this time, instead of this position, we're going to change another position. We're going to go from a GAA to a GCA for the non-template strand, and then the template strand will be CGT. This will make an RNA molecule that is GCA. Looking at the genetic code, we can see that GCA will now make, instead of glutamic acid, alanine. It's okay if you don't know this yet, but alanine is neither positively or negatively charged. Glutamic acid is a polar molecule. Alanine is a nonpolar. So it turns out that this mutation could actually have quite a big impact on how that protein is going to fold. So this is a more severe phenotype compared to the silent mutation. Now there's a special kind of a missense mutation that I'm going to talk about next, and it's called a neutral mutation. A neutral mutation will change the amino acid being made, but it'll make it a similar amino acid. So instead of a GAA, this mutation will now be a GAC. That will make a CTG for the template strand. When this makes an RNA molecule, we'll make an RNA codon that is GAC. If you look at our genetic code, GAC makes an aspartic acid. Aspartic acid is similar to glutamic acid. They're both negatively charged. They're both polar. So we say it's neutral because it changes the amino acid, but it's less likely to have a strong phenotype because it makes a very similar amino acid. Now, don't get me wrong, it could still have a negative impact, but our prediction would be that it would not be as strong of an impact. The last one I want to talk about is when you change the coded amino acid into a stop codon. And so we call this kind of a mutation a nonsense mutation. So you will go from a GAA, and in this case, we're going to just change one nucleotide again, but it's going to be a TAA. That'll make our template strand ATT. As that's transcribed into RNA, the RNA will be UAA. As that goes through translation, there's no amino acid for UAA. It's one of our 3 stop codons. So this will prematurely stop translation. Just like we saw with the frame shifts, it matters where this nonsense mutation occurs. So for instance, if this is our gene here, with our stop here, and our start here, if we make a premature stop codon here, UAA, just at the very end, so you're only missing the last couple of amino acids, this has a less of an effect. But if you introduce that stop codon here, closer to the start site, so the prediction would be that this premature stop codon up here would be problematic, because the finished protein is going to be missing all of this. And so that's not necessarily a good strategy for a good, successful protein. So I'm going to call this just other ways to classify mutations. These are still changing the DNA sequences. In some cases, they could still be missense or nonsense mutations, but we want to talk about them a little differently. The first one I wanted to talk about, and we've mentioned this to some extent earlier when we talked about the FOXP2 gene, but we can have mutations outside of the reading frame. So outside of that open reading frame, the ORF that we've talked about. So if we have our gene here, and we have our ORF here, that makes the protein. Remember, we also have a promoter here. We have these enhancer or silencer sequences down here, and we have other regulatory portions of this gene. It's possible that we could get a mutation here in the promoter, or maybe in one of these other sequences associated with this gene, and that's going to affect the way that protein functions. It could completely turn it off. It could overexpress it, underexpress it. There's a lot of different kinds of an effect, but the sequence of the gene itself is not affected. The next thing I'm going to talk about is just the terms lethal versus beneficial. We often think about mutations having a negative effect, and of course, if you think about a lethal effect, that's the most negative effect. So this is a mutation that kills the organism. It is an essential gene, and when you mutate an essential gene, the effect is often lethality. On the other extreme, a beneficial gene may provide a new function, could be a completely new function for the organism. It may provide an improved function. These kind of mutations are often the driving force of evolution. Sometimes mutations appear in populations, and at some point in that population, these mutations now will have some kind of benefit and allow certain organisms to have a reproductive advantage. Another one I want to talk about briefly here are what I'm going to call conditional mutations. Conditional mutations can be lethal. They can also be beneficial. And the best way to think about conditional mutations is to think about temperature sensitive. And we mentioned this term before, but let's talk about it again. Let's say you have a gene here, a wild type gene, and it will produce a protein that folds like this. And let's say it's 37 degrees Celsius. And if you increase it to 42 degrees Celsius, it's going to fold the exact same way. And so it's going to have the exact same function. However, if you have a mutation on this gene, changes the nucleotide sequence, maybe at one temperature, it folds just fine. But at a higher temperature, like 42 degrees Celsius, maybe it doesn't fold fine. And now it's non-functional. There's a lot of different ways you could look at this. It could be that now it has an improved function. It could now have a lethal function. And I should also state it's not always temperature. It could be something in somebody's diet. It could be something else in the environment. It could be some kind of oxidative stress. There's a lot of different things that can cause these conditional effects. It could be that you have a conditional mutation and you never knew you had that conditional mutation until you experience that condition that leads to this misfolded protein. Conditional effects will always change the way that protein folds, but the condition can vary. The last thing I want to talk about are suppressor mutations. Let's say we have this gene. Let's call it YFG for your favorite gene. Your favorite gene here is now, if it becomes mutated, YFG, still YFG, but now there's a mutation in it that prevents your favorite gene here from doing its job. So we always say, does not do its job. A suppressor mutation is a kind of a mutation that allows this otherwise mutated allele to now do its job. So let's talk about two types of suppressor mutations that allows YFG to ultimately be able to do its job. The first one is called an intraspecific suppressor mutation. An intraspecific mutation will take this cell that's expressing your favorite gene, but it's not able to do its job, and it reverts it. It will either take a mutation, this mutation, and reverse it. So maybe it was an AAA up here, and now it's an AAT here, and now intraspecific mutation takes it back to an AAA. So why not write that down here? AAA, and this might have been an AAT. And now, intraspecific mutation here takes it back to an AAA. And we can all rejoice and be happy because now, once again, your gene is doing its job. Now, it could also be somewhere else on this gene, and another mutation on this gene could now allow it to fold the way it's supposed to fold, even though it has a mutation. So it could be that, as an alternative to this, you might still have this mutation of an AAT, but maybe now there's a new mutation down here that now allows this mutation to be okay. It allows the protein to ultimately fold properly. The other kind of a suppressor mutation I want to talk about is called an interspecific mutation. In this kind of a mutation, the mutated YFG here remains mutated, but now there is another gene here, and let's call this YOFG for your other favorite gene. You can have two favorite genes. And now this other favorite gene becomes mutated. And when it becomes mutated, together these two mutants can coexist to allow that cell, or this original protein, your favorite gene, to once again do its job. Again, an interspecific suppressor mutation is a mutation on a different gene that when it exists with this first mutation, it allows this one to do its job. It's possible that up here, our favorite gene here, has to fold like this to do its job. But the mutation makes it look like this. And maybe for it to do its job here, this other gene, your other favorite gene, has to bind to it. But because of this way it's folding here, your other favorite gene here can't bind to it. But now when you make this other gene here, you may still create an improperly formed YFG here, but now your other favorite gene now has a mutation, and as it's mutated, it can now interact with this to do the job. All right, that is all I have for this podcast. If you have any questions at all, please let me know. If not, I'll see you in class. Bye.